susceptibility to shearing stress. This work is taken from Journal of Plant Physiology and has been studied by Yrryang and colleagues. Today, my talk consists of objective, introduction, materials and method, results and discussion, and the last conclusion. In this study, a group of researchers would like to study the importance of GDP-L-galactose phosphorylase in tomato by Antisense technique. The changing of temperature severely affects growth and quality of plants. When the plants were exposed to high or low temperature, reactive oxygen species are generated. Consequently, the plant cells are damaged. When the plant has a lot of reactive oxygen species, which are unstable molecules, the reactive oxygen species can share electrons from plant cells, so that the plant cells are damaged. At that time, the antioxidant mechanisms are being adapted to cope with shearing stress. Ascorbate is one of antioxidants can share electron into reactive oxygen species to make more stable. Therefore, the reactive oxygen species cannot damage plant cells. Moreover, the ascorbate has other functions in plants. For example, protector, coenzyme, flowering, senescence, and adaptation. The L-ascorbic acid is synthesized in ASA by biosynthesis pathway. Many researchers have been studying on Alamidosicariana, but this group of researchers focus on Solanum glycopersicum or tomato. The GDP in ASA by biosynthesis pathway has been of significant interest to many researchers. Let me enlarge this area for you. And for the GDP in ASA biosynthesis pathway can convert GDP-L-galactose to L-galactose 1-phosphate. In order to investigate the importance of GDP, a group of researchers try to deplete the GDP gene and the ability of tomato plant to tolerate the shearing stress was observed. One way of molecular biotechnology using antisense technique can deplete the GDP gene. The mRNA from SLGDP antisense can match with SLGDP mRNA and inhibit the GDP translation. For the materials and method to create the antisense transgenic plant, the first, the total RNA was extracted from tomato tissue and isolated using 10 column isolation kit. And after that, the cDNA was synthesized from mRNA by MULV reverse transcriptase. And then the cDNA was amplified using tacrymolase. For the cDNA fragment has a overhang and the plasmid vector has T overhang, and then the cDNA fragment was registered into PMD HNT vector and transformed into E. coli. For the colony obtaining recombinant plasmid was selected using standard molecular method. And then for the antisense construction, first, the SLGDP recombinant was extracted from E. coli and cut to be cDNA fragment. And next, the cDNA fragment was amplified using antisense primer. For the antisense, cDNA fragment was integrated into PBI 121 and transformed into E. coli again. And for the colony harboring recombinant plasmid was selected using standard molecular method. And next, the SL-GDP recombinant was extracted from E. coli again and transformed into Agrobacterium tumefaciens. And next, the Agrobacterium tumefaciens was infected into tomato leaf. And finally, the wild-type and transgenic plants were cultured on selective medium and treated at 4 degrees Celsius. And for the analytical method, for gene expression was analyzed using real-time PCR. For the protein expression was detected using Western blot technique, and the subcellular localization was analyzed using transient expression. And for the real-time PCR and Western blot technique, you already studied in the class. Let me describe the transient expression. For the subcellular localization of SL-GGP, GFP fusion protein, and the first, the cDNA fragment without terminal codon was cut and integrated into GFP vector, and then the recombinant plasmid was transformed into onion epidermal cells by gene gun. And for the transient expression was detected using fluorescence microscope. And the next, for the total ascorbic acid, the total ASA was analyzed using 2,2-bipyridyl and folic ion changed to green color by spectrophotometry. And then for the merodialdehyde was measured using TBA changed to red color by spectrophotometer. And the next, for superoxide and ion was detected using NBT changed to red color by histochemical detection. And also the hydrogen peroxide was detected using DAB changed to blue color. And the last, for the APX activity was determined using ascorbic substrate. This slide for the result and discussion, this slide shows the amino acid alignment. This is the amino acid alignment between tomato and different species. There are two highly conserved regions. The NLS is nucleus localization signal, and the HIT is the motive for enzyme catalysis. And the next, this slide shows the RNA and SL-GGP protein gel rod analysis. You can see the mRNA level of transgenic plant were lower than wild type, and also the protein gel rod, the protein content of transgenic plant were lower than wild type, indicated that the gene expression was decreased. And the next, for the subcellular localization, and you can see this is the SL-GGP GFP protein was expressed using NLS signal into nucleus. You can see in the circle. And then this slide shows the total ascorbic acid content. You can see at 6 hours, the total ascorbic acid content of transgenic plants were lower than wild type, indicated that the ascorbic acid was decreased. And this slide shows the MDA analysis. can see the MDA content of transgenic plant were higher than wild type, indicated that the cell membrane damage was increased. This slide shows the APX analysis. From right picture, the APX activity is related in L-ascorbic acid content. From figure 6, you can see the APX activity of transgenic plant were lower than wild type, and this is the hydrogen peroxide analysis. You can see at 6 hours the density of hydrogen peroxide staining in transgenic plant were higher than wild type, and also this slide shows the superoxide and ion analysis. And for the density of superoxide and ion staining in transgenic plant at 6 hours was higher than wild type. And this slide shows the growth analysis. You can see at 6 hours the growth of transgenic plant were lower than wild type. All of the results are summarized in this table, indicated that the tomato chewing tolerance was decreased. Therefore, the GGP gene play important role in tomato. Finally, I would like to thank Assistant Professor Dr. Pikamon Kandit, my seminar advisor, and also thank Associate Professor Dr.